Hello, and welcome to the University of Miami's Rosenstiel School of Marine and Atmospheric Science. My name's Emma Ponce, and I will be your virtual tour guide. Today we're at the Rosenstiel School campus, which is located on the beautiful island of Key Biscayne, and you're going to get a chance to see all the different buildings and some of our beachfront locations. So stay tuned for that, and let's go start our virtual tour. Alright, so right now we are kind of centrally located in our campus. We're right in kind of this main parking area that's kind of in the center of all of our buildings. And our next stop is going to be the dock, where we're going to learn more about our research vessel, the Walton Smith, and also our fleet of small vessels. So in terms of the Walton Smith, like I mentioned, it is a large research vessel. It can hold about 13 scientists and 6 crew members at a time, and there are absolutely opportunities for both undergrads and graduate students to participate in cruises on that boat. So let's go check out the dock and the boats like I just mentioned, and then on our way back we're going to check out our scientific diving training facility. So let's go. All right, so we're standing in front of the university's research vessel, the Walton Smith. It is a large green catamaran, which means that it is mostly a coastal ship. So because of how flat it is, it's able to go super shallow, which is really good for things like coastal work and kind of looking at coastal communities and health there. So let's take a closer look at one of our small boats that we have in our Rosenstiel School fleet. So this is one of our smaller boats. It's relatively brand new. You'll notice that it's wrapped really cool. We have our nice logo, and we also have this tiger shark skin wrapping on it, which is really cool. These boats are used for shorter dive trips, as well as the Shark Conservation and Research Lab. So they also use these boats to go do their shark tagging. Basically any kind of smaller group field work, we're using these smaller boats for that. But I want to take this opportunity to tell you more about my personal experience with the University of Miami and the Rosenstiel School. I actually did my undergraduate degree here at UM. I was a double major in marine science and biology, and I also got a minor in chemistry. It was a busy couple years, but I was able to do really cool things like be on the Walton Smith during research cruises and got a lot of really cool hands-on research experience. I loved my experience so much at UM undergrad that I decided to stay on for my master's. So I did do the accelerated master's degree, the Master of Professional Science. I was in the Tropical Marine Ecosystem track, and my internship was with NOAA Southeast Fisheries Science Center, where I conducted a coral disease project that recently got published. After that, I spent a year in the dean's office doing some outreach and tours and kind of like the stuff that I'm doing right now, which I really enjoyed, and I felt kind of rounded out my career a little bit. During that time, I was always looking to kind of get back into academia and back into science, and I'm excited to say that I'm actually just finishing up my first year as a PhD student in the Department of Marine Biology, studying coral again, but this time with a focus on ocean acidification stresses and kind of OA effects on coral physiology. So I hope you enjoyed that, and feel free to ask me any questions. I'm happy to answer. And so now I'm going to turn it over to Miguel McKinney, who is in charge of all of our boats. He's the director of marine operations, and he is going to tell us more about the Walton Smith, and he's going to give us a tour of the Walton Smith. So let's go.
Hello, welcome aboard the University of Miami's research vessel, Walton Smith. This vessel is, I'm happy to show it to you, it's one of the jewels of the RSMAS campus. Please come aboard, let me show you some things. Right now, we are in what's called the wet lab. This is where a lot of the quote-unquote wet research gets done on the ship. When we take aboard biologicals and things that are generally messy, this is where this aspect of the work gets done. If we're working with things that produce a lot of hazardous fumes, we have equipment that's designed just for that. This is the fume hood. Our chemical lockers are here, and on the ship we are also our own fire department. Okay, let's go into the dry lab. These are 2 of our technicians that have been with us for over 20 years. They handle all the sensors and equipment that the scientists don't bring. This is called the dry lab, and if you want to pan around, please feel free. What Chip and Don are looking at is the equipment that operates the sensor that's on the back deck. Before this video is over, we'll go and show it to you. It's a piece of equipment called a CTD, and they're the technicians that operate that. In this area here, known as the dry lab, although it's nice and clean, we're preparing to go to a dry dock, it is normally chock full of science equipment. It can be computers, it can be sensors, instruments. We've had AUVs on board, and scientists would bring things on board that look like an arcade aviation device. As you can see right here, we have a number of freezers. We have a lot of space for provisions. In the wet lab, I omitted to show you that we also have freezer space for samples. We have ample berths. We take as many as 13 scientists on board. This is the main level. We have accommodations down below as well. And over on this side, we would have the area where the technician resides and has all his sensors and control equipment here. Now let's go into the mess. And thankfully our mess is not messy, it's nice and clean and tidy. This is where all the eating is done, and over here is what's called the galley, which is where all the food is prepared and all the provisions are kept. And also, let's go up top to the bridge and take a look at the controls. The Walton Smith was built with science in mind. The ship was designed and we took delivery of it in 2000. The vessel is owned and operated by the University of Miami. The majority of the sponsors and users of the ship include the National Science Foundation, NOAA, and the Office of Naval Research. We also work with UNOLS, which is the University National Oceanographic Laboratory System. UNOLS provides leadership and guidance to all the national academic research fleet to which we are a part. Now here we are on the bridge. We have state-of-the-art equipment. As you notice, one of the things that's missing, you don't have the old-fashioned control wheel. You have all these computers and sensors for navigation, and also for the captain to be able to monitor a number of systems throughout the ship. The Walton Smith goes to sea on average about 22 cruises per year, and we normally log about 140 days at sea annually. The places we go are quite varied. Although the vessel is designated as a coastal vessel, we are a can-do operation. We have been as far north as New York State, we've been as far west as Mexico, and as far south as the US Virgin Islands. Now that we've seen the bridge, let's go outboard onto the O1 level, where it's the upper level where we sometimes have storage of equipment, extra boats, and that sort of thing. Let's go. Watch your step. Out here, we have ample space for additional equipment. Some scientists bring aboard so much gear that they can't store it inboard. They may have to take what's called a sea container or a Connex box. We have the space and capacity to put it on board here and store it. Our emergency supplies and equipment is stored out here. There are cruises where we do a lot of diving, where we have to get to the shallow spots. Although this ship is built with shallow water research, there are even more shallow areas where divers choose to go. The ship can't go. We would take additional small boats on board and store them back here on the O1 level. What you're seeing here, and I apologize for the covers, but we have the oceanographic winches, and they would use to haul up the scientific packages, the payloads that go over the back end of the deck. Right here is called the winch cab. And from here is where the captain or the engineer or the designated person controls the winches. The ship, this winch cab is capable of controlling the ship from the rear, from aft, and moving the ship accordingly. For medium packages, we're able to not need to go to the yard or to go to some place to crane aboard our gear. And this is where we would do this. Over here, we carry our own other supplies for the ship, including fuel. Some of those small boats I told you, they would use gasoline. We would have our own petrol shed to use for here. And this is essentially it. And up top here on the O2 level is all of our electronics suite. We have an orb, which is a rain gauge. We have other meteorological sensors that would detect wind and speed, velocity, and of course the ship's radar and navigational systems. What we have here is called a CTD. It stands for conductivity, temperature, and depth. And it's a device that's used on virtually every major research vessel around the world. And what it does is it takes water samples. It does more than just that. It's got an altimeter on board and other sensors. It takes these samples at different depths of the water column. And the results from these is taken up through a wire in real time to the controllers on the ship, which you saw earlier, the two technicians. This data is compared with other samples around the world, and it gives us a sense of what the real oceans are really doing, temperature-wise, movement-wise, things like that. We'll turn our attention to this, which is called an A-frame. Basically, this allows us to handle equipment like this, equipment that is heavy and also needs to go over the side of the vessel. If you didn't have this ability to hang things over the deck, you would have them on the deck and have to push them over, which is not safe. And it's also dangerous and for the delicate equipment. What you're seeing here, if you could pan right here and look at this notch is removable. Sometimes you have equipment that comes on board, you might need to hold it dead center above and deploy it. Like for example, oversized equipment or maybe an AUV or an ROV. So this is removable. If you look over here, we have this, it's also a removable notch and it's called the moon pool. Generally, we remove this when we're conducting mud samples. We're doing that kind of work. Over here, what you're going to see here now is what we call the safe boat. This is a rib boat. It's used primarily for the safety of the vessel. It's their day-to-day boat. Sometimes we use it also to ferry scientists to very shallow areas when we didn't bring other small boats. But this is also our emergency boat for the ship.
Alright, so we just finished up an awesome tour of the Walton Smith. We heard from Miguel McKinney, who works in the Department of Marine Operations, and our next stop is going to be the Scientific Diving Training Facility. We're going to get to take a closer look at that and hear more about what kind of stuff goes on in that facility. So let's go. Alright, so now we are in front of our brand new scientific diving training facility, which is behind me, and it's actually the only of its kind in the continental US. So this facility was really designed for the scientific diving course, which we teach here, that is a graduate level course. We also teach a free diving class taught by Dr. Claire Paris, which is again a new class that we're having starting in the fall semester. It really is specifically for these scientific divers to work on their hard skills, their training, so that when they complete the class, they come out of that class with an authorization from the American Academy of Underwater Sciences. All right, so we've checked out our scientific diving training facility. We headed over to the dock and we were able to see the beach and some of our smaller boats and also our larger boat, the Walton Smith. We are now headed to the SLAB building where we're going to check out our library, a couple classrooms, some administrative offices, so let's go. So now I am in the SLAB lobby. To my left is the Graduate Studies office. On my right is the seminar room. So this is a space where students, both MPS, MS, and PhD, give their student defenses. We often will have guest lecturers that come and give lectures in this room. It can seat about probably 75 people. And so this is where we do some of our larger lectures. All right, so we're now in the Dean's Office atrium. This is normally where we start our in-person tours, but I wanted to just kind of quickly mention kind of what goes on in this area. So we have the Office of the Dean. Our Dean of the Rosenstiel School is Dr. Ronnie Abasar, and his office is right over there. We also have our Communications Department. They are extremely active in kind of working with the local news media to make sure that our research is being broadcast to the local community, and also things like social media and stuff like that in terms of communication. And then we also do a lot of outreach. So different individuals and labs will often host school groups, and we're working on making kind of general outreach a more university-wide initiative. And now we're going to check out the second MPS Student Lounge. So let's get in here. And so we have student lockers on the right. Obviously you can leave your bag, your books, whatever you need to during the day. You can rent a locker totally for free and leave your stuff there during the day. We also have a little kitchenette area, just like in the other lounge. You have a microwave, refrigerator. We also have a TV if you want to watch the news or sports. But this is basically a space where MPS students can get together and work on homework, work on assignments, or just kind of relax after their classes. Alright, so we just came from the MPS Lounge, and we also checked out the Graduate Studies Office. And I wanted to mention that the Graduate Studies Office, or GSO, essentially handles all of the paperwork registration for all of our master's students, that's MS, and PhD students. And so they handle all of your applications, your registration for students in all 5 of our departments. So that includes marine biology and ecology, marine ecosystems and society, atmospheric sciences, ocean sciences, and marine geosciences. One of the questions I get asked a lot is what is the difference between an MPS and a MS? So the difference between a Master of Professional Science and a Master of Science. And one of the biggest things is that as an MPS student, instead of working one-on-one with an advisor, you're actually doing an internship. And the hope is that you do this internship, it kind of gets you in the door at an organization that you want to work at. So the idea is for MPS students, this program is really designed for students who want to enter the workforce immediately after graduation, whereas an MS is more designed for students who want to stay in academia and potentially go on to do a PhD, postdoc, and that kind of academic route. So for our MPS students, they're doing internships at local organizations, places like NOAA, Fish and Wildlife, National Marine Sanctuary, organizations like that. So you do an internship and you also write a thesis. So that's another really common question I get is, do MPS students write a thesis just like master's students do? And the answer is yes. So both programs are thesis focused programs. With the MPS program, you just have a little bit more flexibility. So your thesis doesn't necessarily have to be hard data or research focused as it would have to be with a Master's of Science, essentially. I also wanted to mention that 80% of our master's students are part of the MPS program, and 94% of our Masters of Professional Science students have a job within 6 months after graduation. All right, so now we're on the second floor of the SLAB building. This is where most of the Ocean Sciences Department has their offices and lab spaces. So the Ocean Sciences Department covers a wide range of topics, including things like studying ocean currents, ocean circulation, marine physics and marine chemistry, and also air-sea interactions. All right, so we're here at Dr. Kim Popendorf's lab, so let's head in and see what she has to say.
Hi, I'm Dr. Kim Popendorf. I'm an assistant professor here at the Rosenstiel School of Marine and Atmospheric Science, and I am a chemical oceanographer. So I look at the chemistry in the ocean. In particular, though, I think about the role that microbes are playing in controlling the chemistry of that ocean water. So this is phytoplankton and bacteria that are naturally occurring and very abundant in the ocean. And we look at the role that they're playing in some large-scale global processes. So phytoplankton in the ocean are responsible for producing more than half of the oxygen that we breathe every year. So they generate huge amounts of oxygen, and then they're also responsible for making the ocean a net sink, or a place where carbon dioxide goes from the atmosphere into the ocean. And that all comes down to the activity of these microbes. So we use chemical tools to look at microbial processes in the ocean and how microbes are changing the chemistry of seawater, and in particular looking at how quickly they're doing this. How quickly are they producing oxygen, and what are they responding to? So one of the things we look at a lot is nutrients in the ocean. So that's phosphorus and nitrogen. And different microbes are going to respond to those differently. So that's one of the things we look at is how do different types of microbes, different types of phytoplankton or bacteria respond to different nutrients? And how does that vary in different places, comparing coastal ocean to open ocean, to low nutrient areas in the ocean and high nutrient areas? And thinking about also how things are likely to change in the near future, comparing temperature in seawater now to what's likely to be increasing temperature in the near future. So, and then we're trying to put this all in context of these big picture questions of how does this really control large processes on Earth, that production of oxygen and drawdown of carbon dioxide. So we use a mix of chemical tools here. So this is a liquid chromatography, mass spectrometry instrument. We use some other tools for looking at nutrient concentrations. So with these chemistry tools here, we can look at biochemicals within the cells, and that can give us a lot of details about who's doing what in the ocean and how quickly they're doing it. And then we also use a lot of biology tools for looking at those microbes and assessing how many they are and who's there. And we try and bring this all together to really look at these interwoven biological and chemical processes. Most all of our work here, we're collecting environmental samples to do this. So we want to see what's happening out in the ocean. So we go to the coastal ocean, and it's great. We're a few hundred feet from the beach here at the Rosenstiel School, and we use that all the time. So especially with students, we can go out there and sample, run experiments, see what's happening, see how that changes day to day, season to season. And then we also use the Walton Smith from the Rosenstiel School to go out and sample a little further. Again, this is great to be able to take undergrads out to sea and use these oceanographic tools. And for my graduate students, they often participate in much longer scale cruises, 30, 50 days at sea, where they'll do a large transect across the Pacific or across the Indian Ocean and bring samples back to the lab that then we analyze here. And with this too, these are all interwoven questions about the chemistry and the biology. So we work with a lot of other people here at the Rosenstiel School and beyond who have other tool sets to couple with this. So we work with atmospheric chemists and we work with other biological oceanographers and with geologists to really be able to answer these interwoven questions about how the ocean is functioning on a broad global scale.
All right, so we just left Dr. Kim Popendorf's lab. Now we're going to head downstairs to check out the library. So let's go.
Welcome to the Rosenstiel School Library. We're your Marine and Atmospheric Science Library here on campus. We are available to you physically here on campus as well as digitally online, so you can log on to the library website, library.miami.edu/RSMAS Lib. So anything I'm telling you now, you can always find there as well. We are a part of the University of Miami library system, which includes our main campus library, Richter, architecture, business, law, and also our medical library down the downtown campus. Any of the material that is circulating within the libraries is available to you via request. You just have to go online to the catalog, log in with your Canlink login and password, and you can request the material and have it sent here to this library, or if you happen to live closer to downtown, to medical, or over to the Coral Gables campus. Now, if there's material here in the library that we don't have, but you still need for your research goals, go ahead and do an interlibrary loan via our Iliad system. That will let us go out to other libraries and ask for that material and make sure we can get it for you, either in a digital form as a PDF or as a physical item that we can have available to you here in the library. Let me go ahead and show you some things that we've been talking about. Let's go this way. Right now we're walking past our library's collection of expedition reports and, of course, our wonderful underwater photography contest that's displayed here in the library. And for study areas within our library, we have a few different rooms, one of which is our media room. This allows our students to go ahead and display their projects, their reports, have committee meetings, and do a number of different things with study groups to make their life a little bit easier and have a little bit more community within their education. And we have something new in our map and chart room. So the media room is a great space for smaller groups, but what happens if you have a larger group? Well, we have an option for that as well. This is the library map and chart room. So we've used this for thesis and dissertation defenses. We've gone ahead and done seminars, webinars, as well as guest lectures, all within the library here. So there's an ample space for you to be able to come in and do your work individually. Go ahead and practice a presentation if you need to, or go ahead and just sit quietly and try and get some research done just by yourself so your lab mates can't find you and you can get a few more minutes in. This is the general seating area for our library. We've got open area seating here, as well as soft seating to the side if you don't really feel like sitting at a table. Our collection's over on the side here. We are a one-floor library, so all of our material is here within this side of the library. We have our bound journals in the front, but about 80% of our journals are available online, so you just need to go onto the library catalog to access those. We also have all of our books, our monographs over here. Those are all of our books, and they're filed by Library of Congress call number order. If you have any questions or you can't find something, just come and see us. We'll help you out, I promise. We don't bite. We also have study carrels available for our students to sit at and take a look at materials, as well as our current periodicals that are over on the side. There are a few periodicals that are still coming in print form and not online, and those we make sure are available to our students. And if you're ever on campus and you forget your laptop, don't worry about it. We've got Macs and PCs available for our students to use. You just need to log on with your Cane-Link login and Cane-Link password, and you're good to go. And with the Adobe Cloud Service and a Microsoft 365, you have access to all your information if you store it in the cloud. And you also have available to the university digital collections, which are available here on our library webpage. If you go online, you'll go to the digital collections online here. And that's going to give you all the digital collections within the entire university. Specifically for the Rosenstiel School, we have ours here. So we've got an individual splash page if you're looking for information. So we've got studies in tropical oceanography. We've got a photo collection, which is kind of nice to look at, for all the images that have been donated to the university, and they're slowly being cataloged and updated, so make sure you take a look because it's never the same collection twice.
All right, so we're at the MTLSS building. This building houses our sustain tank, also known as our wave tank. It also houses a lot of cool invertebrates that we're going to see soon. And so let's go inside so we can hear from Dr. Brian Haus about the sustain tank. Let's go.
Good morning, how are you doing? Welcome to the Sustain Laboratory. I'm Professor Brian Haus of the Ocean Sciences Department, and I can show you around what we do here and give you a little idea. This, you see the laboratory is up over here, and we have it elevated, of course, for protection of it, but also so that we can get underneath and do work from the bottom. So you see it's up high. There's a lot of space around because we do put a lot of different equipment in here. We never know how it's all going to fit in. Yeah. This laboratory can do some of the highest wind speed measurements in the world, the highest, and it's really large scale, so we can understand what happens in really intense conditions on the ocean. The sustain facility has some unique capabilities. We can put seawater in here, and we can blow a Category 5 wind, which no other laboratory in the world can get to that level. And because of its size, we can do model testing like what you can see behind me here, which is a bridge model to look at, which is outfitted with 60 pressure sensors all over the bottom of that, to look at the uplift load when you have waves hitting the bottom of that, because that's a big failure mode for, that's how a lot of bridges can be damaged during really severe conditions. And so this is work we're doing as a consortium called the HURRY Consortium with our civil engineering department here with, on this problem. The neat thing about the facility is because we have this unique capabilities, a lot of different scientists and disciplines outside of my own, which is physics of the air-sea interface, can do work here. We've done work with coral reef ecologists and biologists to try to understand how reefs contribute to the dissipation of wave energy and can protect shorelines. We've done work, people trying to understand gas transfer in really high winds, because that's an important part of the global climate picture. So there's a lot of different people that can be involved, and it's a lot of different things that we can do here, which is really exciting. I learn something new every day. It's rare that there's a day coming in here that we're doing the same thing.
All right, welcome back. Our next stop is going to be visiting one of the coral labs, Dr. Andrew Baker's Coral Reef Future Lab. So let's go, follow me. Hey Liv.
Hey Emma.
I have a tour for you.
Wonderful. Hi everybody, my name is Liv Williamson. I'm a third-year PhD student here at the Baker Lab, which is commonly known as the Coral Reef Futures Lab here at the Rosenstiel School. And we study coral reefs and how they might adapt or acclimatize to better survive climate change. To just start out, corals are animals. They're related to jellyfish and anemones in the phylum Cnidarians. They have tentacles with a mouth in the middle, and they form colonies of genetically identical individuals that make these big, beautiful colonies that are great structure on the reef. They also calcify, so they take calcium and carbonate from the water and make skeletons, which is what you see basically when you see a coral. It looks like a rock or a tree. It's making a skeleton out of calcium carbonate. And then it also has algae that live in its tissues in a symbiotic relationship. Those algae are very sensitive to environmental conditions and can actually cause the coral to bleach if environmental conditions get too stressful. That means that the coral will lose its algae and basically starve because it doesn't have that source of nutrition anymore.
So what we're studying here in this lab is how can we help corals adapt more quickly in the face of climate change, keep them from bleaching, keep them from being stressed out by these conditions, and basically help find intervention strategies to increase their resilience in the face of these changes. So I'm happy to show you some of the corals that we have here in the lab. We work with a number of threatened species that are native here to Florida and to the Caribbean. Here we have a group of fragments of the coral Orbicella faviolata. That's the mountainous star coral. It's been grouped into these small pores. Corals are really amazing because they're colonial animals that can actually continue growing when you fragment them. Here's a whole colony of a coral called Montastria cavernosa. You can see the polyps, the individual coral animals are each of these little dots. There's a mouth in the middle, and if they're underwater, the tentacles would be out as well. So we work with both adult corals and juvenile corals to help increase their resilience to climate change. My research specifically is working with baby corals, juvenile corals, from the very beginning of their lives, trying to make sure that they take up more thermally tolerant algae, which will keep them from bleaching and allow them to keep surviving and growing and basically adding new genetic diversity to reefs.
This whole first floor of our MTLSS building is mostly the marine biology department and includes the majority of our coral wet labs. So our next stop is going to be the Lirman Lab. We're going to hear from Dr. Diego Lirman about the research that they do, which is mostly focused on reef restoration of local corals. So let's come inside. Hi, Dr. Lirman.
Hello, welcome to my lab. My name is Diego Lirman. I'm a faculty member within the Marine Biology and Ecology department. My lab deals or researches on everything that's attached to the bottom. So we are the Benthic Ecology Lab. That means organisms that spend most of their lives attached to the bottom. So we look at corals, we look at macroalgae, we look at seagrasses. For the past 10 years, we've been focusing on active coral reef restoration. That is, we grow corals both within our tanks, as you see over here, and also within our in-water nurseries, and then we plant those corals onto depleted reefs. So we're working on active coral reef restoration. That is one of the aspects of my research. It's been the most rewarding. So this is some of the research that our graduate students are doing. We're looking for ways to increase the resilience of coral reefs. And one of the ways in which we do that is by stressing coral cells within our tanks, trying to prime their response to stress, and then planting them onto our depleted reefs. So that's called stress hardening, and that's some of the work that my current graduate students are completing. The corals that you see in this tank were all grown from coral larvae. So these are all sexual recruits that we are growing and then planting to increase the genetic diversity of our local reefs. We are working with collaborators within the University of Miami, looking at the impacts of restored coral reef communities for mitigating wave energy. So we're able to restore nearshore reef communities in such a way that we not only recover the ecological services that reefs provide, like fisheries habitat, but also benefiting the shoreline communities and the valuable infrastructure that's found within these nearshore environments.
All right, our next stop is going to be hearing from Dalton Hesley, who's a research associate of the Lirman Lab. So we did just hear from Dr. Diego Lirman, and now we're going to hear from one of his research associates regarding one of their outreach programs called Rescue-a-Reef. So let's go.
Welcome. My name is Dalton Hesley. I'm a senior research associate here at the Rosenstiel School and manage our public outreach program called Rescue-a-Reef. So come on in. In addition to our coral reef research and restoration, we built a community program called Rescue-a-Reef with the goal of advancing our efforts to better restore and sustain our coral reef ecosystems. The way we do that is actually by hosting public community events and expeditions so any individual can get involved with our coral reef research and actually help to restore our coral reefs hands-on, alongside our scientists. The reason being is our coral reefs are so important to both local as well as afar communities. They protect our coastlines, they provide jobs, they provide food, as well as a portion of the air we breathe. So it's important that we not only conserve, but restore those services, and the best way to do that is with your support. So any individual that's interested in learning more about coral reefs and actually playing a role in their restoration can reach out to Rescue-a-Reef to not only come on board with us, but support us locally in the community, as well as follow online to learn more about coral reefs and ways to help.
All right, so we just left the Lirman lab. Our next stop is Dr. Chris Langdon's Ocean Acidification Coral Lab that I am personally a PhD student in. So I'm excited to tell you about the research that we do, and let's check out our lab. Let's go. Welcome to Dr. Chris Langdon's Ocean Acidification Coral Laboratory. You've already heard a lot about what coral is and why it's important. What I want to talk about today is the research that we do in this lab. So basically, we are interested in the effect of ocean acidification on coral physiology. What that means is that with changes in our climate, with things like climate change and global warming, it's also associated with an increase in carbon dioxide concentrations in the water. So because humans are releasing so much carbon dioxide into the atmosphere, actually about 33% of that carbon dioxide gets sequestered into our ocean. And because of that, it actually makes the ocean acidic. And I like to use this example of a bubbly Coca-Cola. So if you picture a soda for a second, something like a brand new Coke, it has lots of bubbles in it. That drink is very acidic, and those bubbles that you're seeing are actually bubbles of carbon dioxide. And so we also learned from Liv that corals are made of a substance called calcium carbonate. It's also very similar to the substance that our teeth are made of. So again, for example, think about if you were to drink nothing but Coca-Cola and never brush your teeth. You're drinking that very acidic drink on your very sensitive, delicate teeth. Your teeth will eventually rot out of your head, right? So the idea is that something very similar is happening in our ocean right now. Not quite the level of, you know, a very bubbly Coca-Cola, but even very small incremental changes in the pH, sometimes even as much as 0.1 or 0.2, can have a huge impact on the health of coral because they're made of that very sensitive substance. And so I actually have a little sample right here. I have a little fragment. This is one of the fragments that was part of our experiment. So basically what we do is we have about 300 fragments of coral that look like this. This is a species called Acropora cervicornis, or staghorn coral, that's local to South Florida and the Caribbean. And essentially what's going on is we have a setup where we have our high CO2 treatment and our low CO2 treatment. So what we're doing is we're putting these corals in stressful, acidic, warm environments. And we're also putting corals in kind of just ambient temperature and normal CO2 concentrations. And what we're looking for is differences in their physiology after being exposed to that stressful treatment for a long time. So all of that physiological stuff that we look at, we are analyzing that in the lab that we're in right now. So after these corals have been sitting in these stressful treatments for sometimes up to 9 months, we can actually take them back into the lab, and you'll notice that these fragments that I was holding before is just the skeleton. So this is actually a dead coral, it's just the skeleton, it no longer has tissue on it. And what we're able to do is basically strip the tissue from the skeleton. And once we have that tissue mixture, we like to call it blastate because we use a process called blasting. That mucousy mixture of blastate is basically what tells us all of the physiology stuff. So we're looking at things like lipid content, how fat the coral is, its chlorophyll content, and these are indicators of how well it's able to perform photosynthesis, how well it's able to kind of survive on its own and make its own food. So that's one of the pieces of the research that we're doing in our lab right now. The other big piece of this is that just like humans, corals are genetically different from each other, and they can come from different parents and have different genetic identities. And we sometimes refer to those different genetic identities as genotypes. And so one of the hypotheses of our lab is that different genotypes have differential responses to stress treatment than other genotypes. And so you've probably heard this buzzword called supercorals. One of the things we're searching for are these supercorals that would essentially be corals that are naturally just better adapted at handling changes in chemical stress and temperature stress. So that's why we're looking at their tissue properties to identify which ones maybe have the highest fat content and the highest chlorophyll. These are things that are going to help us identify these super corals that are just naturally better at handling stressful environments. And then further than that, we hope to work with the Lirman lab to essentially outplant those corals that have been proven to be super corals. We just came from Dr. Chris Langdon's Ocean Acidification Coral Lab, and now we're going to learn about another really cool invertebrate called Aplysia. And so let's go inside the Aplysia lab and check them out. Welcome. You can see we have lots of tanks of Aplysia. Come over here first. So Aplysia are California sea hares or sea slugs. Basically, they're really small when they're first born. So you can see all these little dots in here are basically baby sea slugs, baby Aplysia. In terms of the research that we do with them, it's mostly neurological. So we do a lot of brain research, looking at things like Parkinson's disease, Alzheimer's, aging studies, stuff like that. The Aplysia is actually a really cool invertebrate because it's kind of like the marine science lab rat. Their neurons are very similar to humans, very large, which makes it really easy to manipulate and really a good study organism for this kind of research. And also they only live for about a year, so they're in and out pretty quickly, and they're very docile, so they don't fight us on anything. But let's go to one of these other rows and we can check out some bigger animals. The other cool thing about this setup is that you'll notice these stickers where they basically follow this whole row down. So each of these rows has animals and slugs in it. And what these numbers mean is that every animal in this row came from the same litter, basically. They were all hatched from the same batch of eggs, which is pretty cool. So they're all brothers and sisters along these rows. And as we move towards the back, you'll notice that they get pretty big. So these are some of our bigger animals. They're about a year old. They can get to be about the size of a football. And you'll also notice that they're in their mating pairs. So these slugs are hermaphroditic, meaning they have both male and female sex organs, which means that they can basically take any two slugs, put them in a tank together, and they will generate eggs, which is really cool. I'm looking for an egg bundle. At this point, I don't see any. But this guy down here is very active. You can kind of see how big they get once they've reached sexual maturity. And this red algae that you see in here as well was grown at the hatchery across the street to feed these guys. And it's also partially what gives them their color. In terms of opportunities for graduate and undergraduate students, this lab hires a student every semester. So this could be a good place for you to earn money doing things like husbandry, cleaning and feeding and stuff like that. And then they also welcome undergrad and graduate researchers to kind of perform their own research projects. The reason we have so many slugs is because we also grow and ship them to other institutions to run their own experiments on. And we're actually standing in the only place in the world right now where you can buy a slug for research purposes. All right, we just came from the Aplysia Lab, and our next step is going to be to hear from Danielle McDonald in her Toadfish Lab on the second floor. So let's go. The really cool thing about this building is that all of our invertebrates are on the first floor. So we saw a bunch of different corals. We saw some sea slugs that are also invertebrates. But our second floor is entirely vertebrate. So we have things like toadfish and other smaller fish like damselfish and zebrafish that we do a lot of genetics work on.
Hey, welcome to my lab. I'm Dr. Danielle McDonald, and this is the RSMAS Toadfish Lab. And what we do here in this lab is we study fish physiology, neuroendocrinology, behavior, and toxicology. So if you look over here, this is the toadfish. You can see his cute little face there. I'll take him out of the water. Toadfish are extremely tolerant to a lot of environmental stressors, including air exposure. You can see here, they're super aggressive. So if I put my fingers in his mouth, he would totally bite me. We study them for a bunch of different reasons. So we're interested in chemical communication in fish. We're interested in stress physiology, so what makes fish stressed out? Is it listening to ultra music? Is it pollutants in the environment? We're interested in a neurochemical called serotonin, which is involved in depression in humans, and how it might control blood pressure in fish. And we're also interested in how toadfish are tolerant to different toxicants like ammonia exposure. So you can see we have a whole bunch of big adult toadfish in the lab right now. We get them as bycatch from shrimp fishermen. And also we're just starting to try to breed toadfish in the lab. We're super interested in a hormone called cortisol. This is what goes up even in our blood when we're stressed out. And toadfish will experience the same thing. Of course, stress in toadfish is not the same as stress in humans. We get stressed out by work, or if we're fighting with our friends, or not getting along with our brothers and sisters. Toadfish might experience stress if they're fighting amongst themselves, but also if they're being exposed to environmental pollutants. So graduate students in my lab right now are working primarily on a project that's funded by the National Science Foundation, or NSF. And that project is looking at the role of serotonin and the serotonin transporter, which is the target of antidepressants, on the control of blood pressure in toadfish. And we're interested in this because serotonin affects blood pressure. And blood pressure and the control of blood pressure is very important for a fish's survival to low oxygen levels. And we've already seen that toadfish can survive out of the water. That's essentially low oxygen levels for them. So we're trying to understand that process more. Anything that we learn about serotonin can be applied to human health because serotonin is a very important component of human health. So I'm always looking for graduate students to help me out with that work.
All right, so we just came from the MTLSS building. That is one of my favorite buildings just because it's where the majority of our animals live. We saw lots of invertebrates, and we also heard from some other researchers regarding vertebrates like toadfish. So with that, our next stop is going to be the experimental hatchery across the street where we do a lot of aquaculture research. We're going to check out some of our bigger fish and some of our bigger vertebrates that we have around here, so let's go. On our way to the hatchery, but on our way we're actually going to take a quick stop at the Atlantic Oceanographic and Meteorological Laboratory that's run by NOAA. I wanted to mention this here because there's a huge collaboration that happens between NOAA and the Rosenstiel School of Marine and Atmospheric Science. The Cooperative Institute for Marine and Atmospheric Studies, and that is essentially a joint fund between NOAA and the Rosenstiel School that is able to fund PhD students, fund postdocs, and support different research goals, which is really cool. So there's a ton of collaboration. We have lots of students, both undergraduate and graduate, doing internships, getting jobs at places like NOAA. Most of the research that they do obviously is meteorological in nature and oceanographic in nature as well. So because of that, they also have a separate Southeast Fisheries Science Center that's run by NOAA as well. It's actually just right across the street over here. And so that building handles more of the biology. So they do a lot of marine mammal surveys, benthic resource protection, and stuff like that. This is actually where I did my MPS internship when I was an MPS student in 2016. I actually didn't spend a lot of time in this building because all of our field work was obviously in the ocean. But this is where I did a lot of my data entry, and this is basically where I did the majority of my internship. All right, so we've made it across the street to the experimental hatchery. That's where we are right now. We're gonna actually hear from Dr. John Stieglitz about the research that they do here. He's gonna show you a lot of the fish stocks that they have here that we use for aquaculture research purposes. So let's head in and see what he has to say.
Hi, I'm John Stieglitz, a research faculty member here at the University of Miami. And we're here at the University of Miami Experimental Hatchery, where we conduct aquaculture research. We're going to walk inside and see some of the different fish species that we work with here at the University of Miami. Inside this facility is where we have our primary hatchery operation. So within the hatchery facility, we raise fish from egg to market size, examining different biological properties and performance attributes of the species that we work with. In this tank, we have American red snapper, Lucianus hermitatus, which is an iconic species that we're working with right now as part of a federally funded research project looking at 3 different species actually, the red snapper, the Nassau grouper, and the hogfish, all species which have very good market values. We're trying to determine which one is best from a production standpoint in terms of lowest cost of production, best aquaculture performance overall, and these sorts of attributes. So that's the type of research and development activity we do here at the hatchery facility. In these tanks, we have cobia fingerlings. This is another species that we've been working with for years, the cobia. We work in close collaboration with our industry partner, Open Blue Sea Farms, a U.S.-based company conducting farming operations down in Panama. And this species here, these are almost a month old now in this tank. And so they'll be fully weaned at this point onto a dry, inert diet. And at this point, they're at a size where they can be shipped virtually anywhere in the world. And we'll continue to grow out a portion of these for use in our different research trials that we conduct here at the university. These large tanks that are currently holding the American red snapper will also be used at different times during the year for our larval production runs. So if we need to produce commercial scale quantities of any of the species that we work with, we will typically do so in these large tanks that we have here. We also put in this tank here, you see millions, if not billions, of rotifers being cultured. This is the first feeding prey item that we utilize with virtually all of our marine fish species that we work with. And so through optimizing the nutritional composition as well as the delivery rate of these live prey organisms, we're able to get very high survival throughout the larval rearing process for the different species we work with. So this is our basic water quality lab and live feeds preparation area. We also use our microscopes for larval examination, looking at their growth on a daily rate within this small lab space here. This large recirculating broodstock maturation tank, we have our American red snapper broodstock. Now these are wild caught fish captured off the east coast of Florida, and we've conditioned them to spawn in this tank using environmental conditioning methods. And so these fish spawn virtually year-round and produce eggs that we then take into the hatchery, hatch them, and begin the larval rearing process. Within these replicated tanks, we have a variety of different marine fish species, including juvenile cobia that you see here. Now these are being held here for use in a nutrition trial, looking at digestibility of different feeds at different life stages of the cobia. Additionally, we just concluded a research study looking at use of integrated multi-trophic aquaculture with American red snapper juveniles, whereby we looked at the nutrient uptake rate of a seaweed species that's found here in South Florida that also has a good economic value as a co-product cultured alongside the American red snapper. So the wastewater coming out of the red snapper tank helping to fertilize that seaweed, and that's part of an undergraduate research project and is a good example of the types of research that students can get engaged in here at the University of Miami Experimental Hatchery Facility.
All right, so let's head back across the street to the Rosenstiel campus so we can finish up checking out the rest of our campus. Let's go. This next building houses mostly classroom spaces, a couple offices, and in particular the lab for Dr. Neil Hammerschlag's Shark Research and Conservation Lab. And I also want to point out that we do not have any live sharks in this building or on this campus. It's actually all of their field work is done obviously in the field. They're tagging and releasing sharks, so unfortunately we don't have any sharks on our campus, but we can still check out their office, so let's go.
and I'm Director of the Shark Research and Conservation Program here at the University of Miami. Welcome to my lab. So as part of our work, we studied the behavioral ecology and conservation biology of marine predators, particularly sharks. We have projects underway here off Miami, in the northern Bahamas, South Africa, and the Galapagos Islands. We do various projects to understand how human threats such as overfishing, climate change, and even urbanization is impacting the behavior and health of these sharks and what that means for their conservation and how they interact and control and affect the marine environment. This is a great opportunity for graduate research. We offer opportunities at the PhD level, master's level, and also MPS level. The projects differ based on the degree that's being sought after. For the PhDs, they work very closely with me where they design and implement their own independent study, usually 4 separate studies that are thematically linked. For example, I have a current PhD student that is looking at the effects of climate change on shark movement and habitat use, and ultimately to see if these sharks are responding to changing environmental conditions due to climate variation, and what that means for example, their use of marine protected areas. I have a whole team of great master students that are doing a variety of different projects. Some of them are working in South Africa, for example, using drones to study interactions between great white sharks and seals. I have students that are working in the Galapagos to study how nursery habitats, inshore mangrove ecosystems affect the health and survival of small juvenile sharks. And we also have MPS students working on very practical projects that help our whole lab. Some of them are very focused on helping us design field protocols for our research, both abroad and locally. What's also really important to know about our lab is we're very engaged in public outreach. Part of that, we bring people from the public, mostly school children, out with us on the boats or in our labs to get hands-on practical experience in marine biology. The goal of this is to provide these people with opportunities in science so that they get an appreciation for science, technology, engineering, and math, hoping to inspire them, especially the children, to become advocates for the environment while appreciating the scientific fields. So if you want to get involved in the work we do, you have to realize there is also this engagement with the public. So if you like research, if you like outreach and work that's all centered around conservation, biology, well then this is a great place for you.
So this hallway actually used to have a lot of lab spaces in it, but we recently built the MTLSS building, so a lot of the labs that used to be in this hallway have now moved over to the new building. All right, I want to take a second to point out our print shop. It's obviously closed right now, but during the semester it is open all the time. If you ever need help printing things like business cards, if you want a bound copy of your thesis, if you're going to a conference and you need a poster printed, they can do all of that for you right in-house. They're amazing. I've worked with them a ton, and they're extremely helpful in terms of getting posters for conferences and all that kind of stuff. So let's move on. Down this hallway, we have our dive office on the right. Actually, in the next couple months, this dive office is going to be moving to a new location, but for now, it's right here on the right. You'll also notice that there are signs kind of left along this hallway that indicate different classes, different lab groups, and often what will happen is equipment will be left out for those students. So you probably saw the green Pelican cases, those green suitcases over there. That's actually emergency oxygen, which is required to bring with you on a boat whenever you dive. So basically different labs can reach out to the dive office, fill out a dive plan, and then the dive office supplies them with the gear, the tanks, and all the safety equipment that they need. While we're here, I also want to point out our underwater photo contest. So these are some really cool photos, all taken by students and faculty and staff. We host this contest every year, and the first prize is actually an all-expenses-paid cruise, which is really fun. I think it's a scuba diving heavy cruise. And you can see all the different types of photos that are taken. Every year we have different categories that include things like macro, black and white, and kind of different categories that you can submit your photos to. And so this hallway is kind of lined with all of those previous winners' photos. Also, while we're over here, I wanted to point out Dr. Jill Richardson's office. So Jill Richardson is the track leader for the Marine Mammal Science track of the MPS program. She's extremely accessible, and so her office is right here if you ever need to come see her. You can always reach out to her, and basically this is where her office is. I also want to point out Dr. Kenny Broad's office as well. So Kenny Broad is involved in the ABBA Center. He's also worked with National Geographic. He's done a lot of really cool stuff and has a lot of really good connections that students utilize for finding internships, finding job opportunities and stuff like that. So again, his office is over here. And over here we have Miss Maria Estebanez, Dr. Maria Estebanez, who is the track leader for the Marine Ecosystems and Society track. Again, she's super accessible. This is essentially where her office is. She tends to be here very frequently. But essentially this hallway is a really common hallway for different faculty members. So if you're looking for someone in particular, it's very likely that they're in this hallway. And I also want to point out one last thing, which is Fritz Hanselman's office. So Dr. Fritz Hanselman is the track director for underwater archaeology. All right, so we're just leaving the south side of the Growth Center building. Now we're gonna head over to the auditorium, so let's go. You'll notice we have one of our small boats trailered over here. At this point, our field work is relatively limited in terms of the number of people that we can have on a boat at one time. So because of that, we've actually trailered some of our boats. We brought them out of the water. And so that blue water boat is one of the boats that we do use for our smaller dive trips. And the reef restoration and stuff like that. So up here is part of the CIMAS building. It's the Cooperative Institute for Marine and Atmospheric Studies. Right now it's currently being headed by Dr. Ben Kurtman, and they do a lot of really good stuff with atmospheric science. They're able to fund PhD students and master's students, which is really cool. So just another kind of aspect of the research that we do here. So the auditorium is able to hold, I believe, about 300 people. At this point, we are working on ways that we can kind of redesign the auditorium and restructure the auditorium so that it can be used for student classrooms, keeping social distancing in mind because of the amount of space that's in there, which we're going to see in a second. So directly across from the auditorium is the Marine Science Center. This building has a couple of classroom spaces. It also has our salt waterfront restaurant, so let's go check it out. Okay, so our next stop is going to be the MPS Student Lounge. Let's make sure my card works in here. So this is what we call the MPS Student Lounge. This is basically a space for MPS students to get together and work on homework. You have some cubbies over here that gives you room to leave your book bag and stuff like that. We also have a mini fridge if you want to bring your lunch. You can keep it cold in there. As you can see, we have desks and tables and spaces for you to do your homework and kind of get together in groups and do your work. So this space is actually only for MPS students. And the reason for that is because MPS students are here so quickly, right? So they're in and out usually, minimum of 1.5 years. So because of that, they often aren't working directly with an advisor, and they often don't have an office. So this is basically the MPS student office. It's a space for you to do the work that you need to do. We also do have a TV in here. You can watch the news, sports, stuff like that. And this is actually one of 2 MPS student lounges that we have on this campus. Now we're gonna leave the MPS student lounge and we're gonna check out one of our classrooms. Right across the hall here. Alright, so this is is one of our newer classrooms. It's also one of our biggest classrooms on campus. It's able to hold probably up to 40 students at a time. Obviously, in the future, coming fall semester, this will be kind of reorganized and restructured to account for social distancing, but it's really nice to have a big classroom space like this that can accommodate students that are kind of sitting 6 feet apart. You obviously have your comfortable chairs, you have your outlets, and we have screens up there where the professor is able to project, the lecture or whatever it needs to be. So yeah, this is one of our bigger classrooms. All right, so we're now in the Cooperative Institute for Marine and Atmospheric Studies. We're going to hear from one of our third-year PhD students, Ms. Kayla Besong. She's going to talk to you about the research that she does that's focused on the atmosphere and climate. She's also going to mention our Marine Science Graduate Student Organization, of which she is the Sustainability Chair and also manages our local garden. So let's go hear from her.
I am Kayla Besong. I'm a third-year PhD student in Dr. Kerben's lab. I currently study a phenomenon known as atmospheric blocking. It's kind of like a traffic jam in the atmosphere. Other members of our group also study subseasonal phenomena such as the MJO and low-level jets. We also study phenomena ranging up to interdecadal timescales. So from subseasonal to interdecadal timescales, our group studies and tries to understand the past and the present so that we can better predict and prepare for the future. We have a long and resourceful amount of computational resources that help aid in our climate forecast models that we use to help understand these types of things. A lot of members in our group are also engaging in a lot of outreach. We're members of the general climate group involved at RSMAS, which also goes to schools as well as gives outreach opportunities right here at RSMAS. We also engage in different blogs such as Subseasonal Chaos, which our group uses to talk about subseasonal forecasting and better get our message out there to the public. Additionally, most of our members are also part of the Marine Science Graduate Student Organization, or MSGSO. This helps get the voice of the students to such as the administration and make changes here on campus. As Sustainability Chair, my mission is to help make our campus more sustainable through things like celebrating Earth Week, gaining awareness, and even having cool projects like the community garden. Hey guys, here I'm going to show you our garden that we worked on. Here we have our sign, our Garden by the Sea. That's a Instagram handle you can go ahead and follow with us. So here's our garden. We've transformed the space to kind of like an empty looking side of a building. It's the perfect place to basically grow, right? So we have a little bit of an iguana problem, and so this is our very marine science student solution to our iguana problem, which helps keep these iguanas out and from eating all of our plants, right? So here I'm pretty sure we got some squash growing over here. We have more squash over there. Peppers too. We have a lot of peppers, and also our basil is thriving, right? So over here is where we mostly had our iguana problem, but this is open to all of our RSMAS community. It's a community garden for people to come collaborate and, harvest from, grow together. We also have a lot of tomatoes growing. As you can see, they're almost ready to harvest, which is awesome. This is great. These are some new additions that we have growing. And again, as you can see, we're all growing this right by the sea.
Next stop is going to be our Salt Waterfront Restaurant. Let's go check it out. All right, so here we are in our Salt Waterfront Restaurant. To my left, behind this wall, is where the hot food is. It's obviously closed right now since we're closed for the summer. But this is where you're able to get your food. Unfortunately, this is an independently owned restaurant, so it doesn't work with the meal plan. But it's still great food for lunch and for dinner. On my left over here, I also want to point out our bar that we have on campus, fondly known as the Wet Lab. Obviously, they are very strict with IDing, so it's generally just graduate students that attend this bar and their faculty as well. But I wanted to point out how much of a great networking opportunity this is. So it's a really great way to kind of have a casual conversation with your faculty over a beer at the end of the day. Sometimes it can be just a little less daunting than approaching them in their office hours or kind of in their lab spaces. So next, I want to take you guys out to the patio and we're going to check out the waterfront view.
Hi, I'm I'm Kelsey Malloy, and I'm a third-year PhD student in atmospheric sciences. I myself study climate forecasting, so forecasting between the sub-seasonal to seasonal time ranges. And I'm also the lead coordinator for Students for Students. Students for Students is an outreach program that specializes in graduate student presentations designed for communities that otherwise would not be able to come to the Rosenstiel School campus. So these are presentations that are catered for specific age groups, so anything through K through 12. And this is really unique because these students can create different presentations on a variety of topics, so ranging from climate change to fossils to fish. Anything within the marine and atmospheric science area. We started this program back in fall of 2019, and we've already done dozens of presentations, and we're able to communicate and engage with the communities that otherwise wouldn't come to this campus. And it's just a really, really great opportunity for our graduate students to get involved in that way.
Hello, my name is Quentin Lawton. I'm a second-year PhD student here at the Rosenstiel School, and I am in atmospheric sciences. I'm also the team leader for our outreach group here at the atmospheric sciences called Canes on Canes. And we are a group of graduate and undergraduate students here at the Rosenstiel School that study hurricanes, and we do outreach events to educate the public about the science of hurricanes and hurricane preparedness. We are really passionate about sharing the knowledge we've learned about hurricanes with the public, and also educating the public on what they can do to prepare for these systems. For Canes on Canes, we've done a number of events to reach our local community as well as the community at large. So for example, recently we had a webinar series on the 2020 hurricane season in the Atlantic, where we shared information on the season and the outlook for the season, and also, of course, hurricane preparedness. We also tend to go to a lot of schools and local events. So we don't have an actual set age group. We like to work with anyone from any age. So we've done things such as going to K through 12 schools in the local community and talking more about hurricane outreach. We've done things at local science museums. We've gone to sailing events. So basically anywhere that there are people here in the local South Florida area, we have a presence.
This is arguably one of the best views in Miami. Just also, a constant reminder of why we're studying what we're studying here, right? We get a lot of really cool wildlife. As you can see, the water is pretty shallow over here. During tours, I've seen animals like sharks, turtles, birds, iguanas, dolphins, all kinds of stuff right in our backyard, which is really cool, and we're very lucky to have such a beautiful space. I want to thank you all so much for joining me on our virtual tour today. For more information, you can check out rsmas.miami.edu, and I look forward to answering your questions.